I am P.Kalaimagal, Assistant Professor, Department of Zoology, Joseph Arts and Science College, Tirunelveli. Dear student, today we are going to study about cell cycle. What is a cell cycle? The cell cycle is a sequence of cell growth and division. This is very important for you. The cell cycle is the period from the beginning of one division to the beginning of the next. The time it takes to complete one cell cycle is the generation time. Cells division involves mitosis and cytogenes. Mitosis involves division of the chromosomes. Cytogenes involves division of the cytoplasm. Mitosis without cytogenesis results in multinucleate cells. These are cell cycles. Next one, eukaryotic cell cycle. Beginning of one division to beginning of next stages in eukaryotic cell cycle. This is cell cycle two phase. Interphase, another one M-phase. Interphase, the longest phase. This is three types phase: G1 phase, G2. This first gap synthesis, second gap phase. Next M-phase. This is mitosis, cytogenesis. Human being 24 hours the cell cycle. This is 95% on the interphase. Next 5% only M-phase. You can see this picture is simply identified and understand. This is interphase, G1 growth phase, post mitotic gap phase. This function ATP, nucleotides, amino acids, RNA, protein synthesis, growth of nucleus. This is longer, two types, longer in non-divide cells, for example human nerve cells, shorter in dividing cells. Next one S phase means synthetic phase. This is DNA replicated. Next DNA replication, histone synthesis, nucleus growth, replication of chromosomes. DNA only replicated. Centriole replicate animal only. Next one G2. G2 phase, pro-mitotic gap phase. This is RNA and protein synthesis. DNA supercoils. Next one mitosis, M phase. This is 4 phase, very important. First one prophase, metaphase, anaphase, telophase. Last one, this is small phase, cytogenesis. Okay. Next, these are small identified. First one gap phase 0. Quiescent arresting phase, where the cells has left the cycle and stopped dividing. Next one interphase, the 3 phase, already you know. Gap 1 synthesis, gap 2 G1. This is cell increase in size in gap 1. Next one, yes, DNA replication occurs during this phase. Next gap 2 G2, during the gap between DNA synthesis and mitosis, the cell will continue to grow. Next one, cell division. Mitosis M. Cell growth stops at this stage and cellular energy is focused on the orderly division into 2 daughter cells. Okay. Next one, middle of mitosis ensure that this cell is ready to complete cell division. This column is very important. Easy identified the cell cycle. Next, the chromosome become duplicated during interphase. Cells are very active during interphase, synthesizing biological molecules and growing the G1 phase. The S synthesis phase is marked by DNA replication. The G2 gap phase occurs between the S phase and mitosis. Hereditary material is passed on to new cells by mitosis or meiosis. Cell division, growth and development, then interphase, mitosis, cytogenesis, and meiosis. Okay. Next one, cell division. Chromosomal packaging of DNA allows efficient distribution of genetic material during cell division. Life cycle requires two distinct types of cell division process. Mitosis and meiosis. These two types only life cycle. Cell division. One cell becomes two cells during an organism's life cycle. Then mitosis. Mitosis is the division of chromosome in a cell, usually followed by cytogenesis or cell division. Daughter cells produced after mitosis have same number of chromosomes as original cell. Purpose of mitosis is replication of somatic cells and asexual reproduction. Somatic cells include all cells in the body except the egg or sperm. Okay. Mitosis is nuclear division plus cytogenesis and protects two identical daughter cells during the following steps. Already Prophase, Metaphase, Anaphase, Telophase, this four phase. First one, Prophase. This is Prophase. During this first mitotic stage, the nucleolus fades and chromatin condenses into chromosomes. Each replicated chromosome comprises two chromatids, both the same genetic formation. This is pair of centrioles. This aster chromosome and consisting of two sister chromatids. This is centromeres. Next one, metaphase. Metaphase of the nuclear envelopes break down. This is the nuclear envelopes break down and a large protein network curled in the spindle attaches to each sister chromatid. These chromosomes are now aligned perpendicular to the spindle in a process called metaphase. These spindle fibers, this is metaphase plate. This is breakdown and a large protein network which attaches to each sister chromatids. Chromosome align on equatorial place due to differential content of chromosome spindle fiber. Next one, anaphase. This is daughter chromosomes. The centromere of each chromosome separates first and moves towards the poles this side. Chromosomes break as centromeres move to opposite ends to the cell. Centromere splits between centromere interzonal fibers formed. Spindle fiber condense to do full chromosome in opposite poles. Okay. This opposite poles same break. This anaphase. Next one, telophase. This chromosome lose their identify and become a mass of chromatin. The nucleus will be reorganized from the chromatin. The cell plate will divide the cell into two daughter cells. This you see, two daughter cells. Cleaves burrow. This reversal of prophase, nuclear membrane, nucleus reappear, chromosome descendants to form chromatin fiber. This is spindle fiber deassociate. This is telophase and cytogenesis. The cytogenesis division of cytoplasm usually occurs between late anaphase and end of telophase. Okay. Next one, significance of mitosis. Essential of normal growth and development of living organisms. Development of a zygote into adult organism. It gives a defined shape to a specific organism. It also helps in repairing of damaged parts and regeneration. Next one, a sexual vegetative reproduction. Number of chromosomes is maintained. Okay student, so in this video you understood. Thank you.